All right, everyone, let's go ahead and get started. Welcome, everyone, to the Center for Reproducible Biomedical Modeling's monthly seminar series. In this series, we're bringing you tools and techniques to help assist you with biomedical modeling. Today, we'll hear from Dr. Joe Hellerstein and Dr. Daniel Weindall about tools they're developing to support parameter estimation in systems biology modeling. We hope you also join us for some of the upcoming months to hear about tools for annotation and visualization of your models as well. For today's session, we're going to have two 20-minute talks followed by about 20 minutes of discussion. So please do hold your questions until after both speakers have shared their presentations. And please do mute your microphones during the talks. You're free to post in the chat if you have some questions that you want us to read off later as well. Our goals for the seminar are really to identify challenges that we all share in our modeling experience and to share our own personal experience and brainstorm new solutions, and ultimately create a more collaborative community of modelers. So hopefully you'll have someone that you can reach out to if you are in need of support. For the discussion session, err on the side of commenting. Many of us likely have faced similar challenges and might be able to offer either useful advice or expand upon your ideas. And again, honest debate is encouraged, so be respectful of everyone. And also please, again, mute your microphones when you're not commenting. All right, so now on to our speakers for today, Dr. Joe Hellerstein and Dr. Daniel Weindall. Our first talk will be by Dr. Joe Hellerstein. Dr. Hellerstein is a senior data science fellow and research scientist at the UW eScience Institute and also an affiliate professor of computer science and engineering at UW. Dr. Hellerstein has also extensive industry experience, having worked in software development for Google and Microsoft, and he brings this experience to the systems biology community to make modeling more robust using software engineering principles. So today, Dr. Hellerstein will present on a Python library that supports parameter estimation, SBstoat. Welcome, Dr. Hellerstein.
Okay, thank you. And let's see. Okay, so now I should be able to share. Great. So I put the link to my slides in the chat in case you missed something or want to go back. Let's see here. There we go. All right. Let me minimize this. Okay, good. So I'm going to chat a little bit about this library, but actually probably close to half of my talk is going to be about sort of setting up some context, giving you a little bit more perspective on parameter fitting. I'm not sure how much of this community is familiar with it, or that the folks who are on this particular seminar. So I want to make sure we're all on the same page, and there will also be maybe some new insights for folks who have been engaged with parameter fitting, but slightly different perspective for thinking about it. So. Okay, so what is it? The sort of basic stuff. This is a model that has been sort of an interesting model on glycolytic oscillations in yeast. I'm not going to go through the details of this. I just want to motivate the reasons for parameter fitting is that almost all these models, when you're looking at time course simulations, mechanistic models, with reaction networks, there are a bunch of parameters that you maybe have a sense of the range. You're not sure about actual values. Kinetic parameters are common. If you have Hill equations, there are probably exponents in there. And so these are what we mean. These are the parameters we're trying to estimate. And the way we do this is we look for how we can find values of these parameters that are sort of semi-known or maybe not known at all, that will make the resulting simulation model consistent with the experimental data. So I'm not going to go into a lot of detail here. This is one model I use as a running example. This is more of an eye chart. You can see there are 10 reactions here, and it's a non-trivial model. There are about 16 parameters, 17 if you included the input flux over here. And just as an aside, I'm not going to be looking at the chat while I'm talking. So Veronica, please interrupt me if there's something that is going horribly awry, like my screen disappears. Okay, so here are the set of parameters that are present. They're drawn from various sources. They're credible sources, but still, the actual appropriate values are not necessarily clear unless you have empirical data to compare it with. The simulation results in some interesting set of oscillations for the various metabolites and glucose. And then the parameters themselves are sort of sprinkled throughout this model, if you can see these boxes throughout here. So it's sort of a complex thing to sort out where all they go. So when we talk about parameter fitting, there's sort of a generic structure to this that we're going through. We have a model like that model I just showed you, the glycolytic oscillation model. It could be in CellML, it could be SBML, it could be a lot of things. And we identify the parameters we want to estimate. We may not necessarily need to estimate all the parameters, just some of them. And then we have some observed values obtained somewhere. I mean, ideally it's wet lab. There may be other sources for this information. And what we're going to come up with are a set of estimates, basically assignments or settings of values to the parameters that we chose, that we felt that needed, we were uncertain about. And the steps are pretty straightforward, sort of like what you would expect. You know, you come up with a new setting of parameters, you run a simulation with those parameter values, then you look at the difference, or what's called the residuals, between the observed values that you obtained that you felt were representative of real world experience and the simulation results. And then you're looking to see if these residuals are small. Typically the sum of the squares of the residuals is used, because residuals could be both positive and negative. And if these are smaller, the settings that you chose, then you say, okay, this is better. We're getting closer. We'll assign this as our new settings. And if you're close enough, if the residuals are really small, you stop, or you run out of time, one or the other. Typically in this algorithm over here, the thing that varies is this part right here, the search algorithm. So this might be gradient descent, it might be differential evolution, it could be a lot of different algorithms, and that's sort of what varies. All right, so there are a few concerns about parameter fitting in general, especially for small data sets, which is very common in biology. And this is sort of courtesy of John von Neumann. Various other people have recognized this, that if you have enough parameters, you can fit almost anything, as you saw from that model, that Wolf model of glycolytic oscillation. There were 17 parameters, so you could fit a lot of stuff. And that's the problem. That's sort of like a core problem in systems biology, is basically, are we in essence overfitting? We have so many parameters and such modest amount of data, are we overdoing it? This is a fundamental issue that limits us in terms of data we have available. I don't know that we can improve on the state of the art there to much great extent. There are some techniques, sort of statistical techniques, that help, something called cross-validation that could be helpful in this regard, but it's something we always have to keep an eye on. The second concern is computational complexity. And so one choice for that search algorithm is exhaustive search, where basically you consider all values or a certain range of values for the different parameters. So we had 17 parameters before. They might have different levels or settings that you consider, and then you could explore the space of possibilities. So here's a simple analysis doing this. Suppose that each simulation, so an evaluation of a single assignment of parameters, single setting, let's say each one of those takes a microsecond. And we're going to talk about, for different combinations of a number of different values for a parameter and the number of parameters, how long it will take. And my scale is going from seconds. So this is in log base 10 of hours. So an hour will have a value of 0 because it's 1. Seconds are obviously less than 0 because it's smaller than an hour. And the scale goes up to the age of the universe. So we've got quite a range here, sub-second to age of the universe. And here's the observation. So this is actually not with a lot of parameters. And there are many models that will have 30 or even 50 parameters in them. And if you look at the number of settings, or number of different levels or values for the parameters, you can see you quickly can get to time required to do these simulations that is on the order of the age of the universe. So obviously exhaustive search doesn't work. You have to be more systematic about your choice of algorithm. Okay, so Here's one approach to thinking about this, about what algorithms are better. Many people explore this. Just try. There are a bunch of different algorithms, for example, in SciPy that you could use. But at the end of the day, what this is really about, it's pretty much akin to you're going on a trip and you want to try and figure out what kind of car is most appropriate. Can I drive my Prius that has very low clearance, or do I need a four-wheel drive? Well, am I crossing streams? Am I going over mountains, or just have highway driving? And that's what this idea of the surface is, the fitting surface. So what this is going to be is this is the sum of square of the residuals for different setting of the parameters. So for example, consider a really simple model. We have a single chemical species. These are both fixed species, so they do not change. So it's just a one variable system, and we're using mass action kinetics. And here I'm plotting out this fitting surface. So the true value is at the red dot, where K1 is 1 and K2 is 2, and drawn from the model over here. As we move away from that dot, looking at the scale over here, you can see that the residuals get larger. So, especially up towards the corners here or down here. One thing you may notice is there seems to be a line of unit slope that seems to be a little bit lighter. And actually, if we go zoom in a little bit, it actually really is there. And that's because this is, of course, a linear ODE. Its solution, the steady state value, will actually be the ratio of these two, of K1 and K2. And so as you go out further time, the sum of square of the residuals is going to be largely due to, did you get the right steady state value? And so that's why it tends to be lower over here. This particular fitting surface is actually pretty easy to figure out because it's what's called convex. And that is that I can pick any point here and basically go down the hill, use gradient descent to get to the point where I get to the red dot. And so that's a relatively nice surface to go through. But of course, that's not, the real world is not quite so nice. If we go back to this example of the glycolytic oscillations, um, we have a much more complex fitting surface. You can see here the red dot again, it indicates the true values. I'm only looking at a combination of 2 of the 17 parameters. Obviously, I'd have to visualize in 17 space to be able to do all of them. So I'm just fixing all the others at their true values and varying these 2 over here. But what you can see is there are ridges throughout here. And so the true value is here. But if I picked up on, if I had the misfortune of starting my search right over here and I was doing gradient descent, I'd probably get stuck in one of these valleys. Now I can use other algorithms like simulated annealing. I could use differential evolution. They'll sort of pop around through this space, but they're not based on really an understanding of the structure of the surface. And so as a result, what you come up with is, sort of dependent on the luck of the draw. Of course, if you spend more time, you potentially can do better. Okay, so to summarize here, this first part of the talk, and that is, if you want to look at the issues affecting complexity and also quality of the fit. Okay, there's the fitting surface. Do you have a convex surface, or do you have something more complicated? The number of parameters, the number of levels you're dealing with, fewer parameters, it's a less complicated space. You can do better that way. You can go somewhat exhaustive. Where you happen to start your search makes a difference. The search algorithm obviously will make a difference too. So all of these factors come into play. And so my net on this, and this is sort of the prep for what I'm going to introduce with SBstoat, is that fundamentally fitting is a kind of exploratory data analysis. I think we can do better than current state of the art, and there are probably some better algorithmic choices we can make. But the bottom line is that the computational complexity is inherently so massive that whether we're increasing parallelism or we have a little more sophistication in the search, it's still going to be something that ultimately is an exploration. Okay, so on to SBstoat, which was what we advertised as the topic for today. SBstoat is a Python package. It's available on pip. There is a Jupyter tutorial. You can, if you look at the slides, you can go to this. One of my to-dos actually is to improve the documents and use, have more and have something, develop something and read the docs. But this tutorial in Jupyter Notebook will certainly get you off to a very good start. So SBstoat, like any fitting algorithm, the inputs are you've got to have a model, you've got to have some experimental data, you got to figure out the parameters you're going to be working with. So here's your experimental data, and you do just CSV files or variations on that. It could be a spreadsheet as well. And then to do a single fit, it's pretty straightforward. So there's the Python import after you've done the SBstoat install. You have as arguments the model, the path to the observational data, and then a list of the names of the parameters, and you just say fit. And you could also, if you prefer, it maybe you don't want to be able to, you don't want to fit to actually all of the chemical species of the model. So there are variations on this as well. Maybe if you had 3 species, you only want to look at 2 of them to do your fit, or you don't want to look at all the parameters. You can figure out subsets as well. And a fitting report will give you some information about the result of the fit. In particular, you can look at the chi-square. You can see the actual parameter values. Typically, since this is, as I said before, really an exploratory analysis, you really need to look at plots to be able to explore your fit. And SBstoat provides a variety of plots. For me, my experience has been the plot of observed versus estimated values is sort of the best. So the dots here are the observed values, the solid line are the estimated values from the simulation. It gives you a feeling that for this particular model, which was this linear chain over here, we're doing pretty well on the first and the last species. Some of the interior species we tend to not do quite so well. Whether that's a result of the fit or the variability in the data is still a bit of a question here. You can see that the scales are somewhat different because these interior species here tend not to have such extreme values because of sort of the way the transformation of the chemical species happens in between. So with lower numbers and the amount of variance in here, you tend to get a little less quality in the fit. You can look at residuals over time. There's a whole suite of plots that are available in SBstoat and is discussed in the in that tutorial. One of the things that is extremely useful when you're doing a fit is to be able to estimate the variance of the fitting parameters. And for this, well, actually, I guess I'd have to reshare. So I guess I won't do the, I was going to do a little demo, but I realized I would have to reshare my screen. Don't want to take the time to do that. Let me just show you what you get from this. So what bootstrapping is, it's a way of essentially generating synthetic data by randomizing the residuals from an initial fit. And then you get new data, and as a result, you can get many more estimates of the parameters, and from that, you derive distributions of the fits. This is something that is quite computationally intensive. SBstoat actually is implemented using multiprocessing. So it allows you, these fits can be in parallel. There's a little progress bar that you see the results of here. It shows you how many bootstrap iterations were done and the time it took overall. In this case, it was 10 seconds to do 2,000. And then the basically the number of iterations per second. And then you can get from this, various results. The standard deviation, that's the primary thing you're interested in for the fits. And then various plots showing again, the interactions between the parameter estimates, which is sort of useful on doing this kind of analysis. I referenced, just sort of keeping track of time, I referenced cross-validation at the start. This is a statistical or computational method for assessing the quality of the fits that you're getting. Basically, you're leaving out part of your data. You do a fit, and then your evaluation of quality is done by applying those fits to data that you left out. And SBstoat supports cross-validation as well. This is also somewhat computationally intensive and easily parallelizable. So each one is what's called folds, the things where you've left out some of the data. Essentially, you're using just subsets of the data. The folds are done in parallel. so that you could then get that information as well fairly quickly. The last thing I'll mention in terms of capabilities is what is suites of models. So you have a set of models that are related because they're actually talking about the same organism or same pathway or circuit of interest, but under different conditions. It might be different initial concentrations. It might involve gene knockouts. It could be a variety of things. And so what you want is to estimate one set of parameter values that apply to all these model variants. And SBstoat provides this too. And you can see the inputs are, you have to specify the models that you're using. Each model has its own set of observational data and potentially set of parameters, although this is a set of parameters and these sets presumably overlap. And those have to have the same value for the models in order to consider a quality fit. So in terms of moving forward, actually this is a forward pointer to the next talk. So besides the documentation I mentioned before, I'm very interested in exploring PETab as a way of providing a tool that will be more generally usable for the community. And so with that, here are my acknowledgements. And I think I'm about on time, maybe a minute or so early. Let me stop sharing.
Thank you, Joe, for a really excellent talk and sort of an overview of the innate challenges in parameter estimation and also a demonstration of SBstoat. Just as a reminder for everyone, we'll have our second talk before moving on to questions, but do feel free to start adding some in the chat if you would like to. I'd now like to welcome Dr. Daniel Weindall. Dr. Weindall is a postdoctoral fellow working in the data-driven computational modeling group of the Institute of Computational Biology at the Helmholtz-Zentrum München. He has worked on several open source software tools that make simulation and parameter estimation of signaling network models more efficient, in addition to his research analyzing complex dynamical systems like cancer signaling and cellular metabolism pathways. Today, Dr. Weindall will present on a data format for parameter estimation, PETab. Welcome, Dr. Weindall.
Thanks for the kind introduction, and also thank you for inviting me. Let me share my slides. There we go. Yeah, so I will present PETab today. It's a format for interoperable specification of parameter estimation problems in systems biology. We already got a great introduction into the topic. That's very helpful. I also like the plans to support PETab in SBstoat, to start with that. I'd like to acknowledge already many people who invested quite some time in the development of PETab. Wouldn't have been possible with the help of all of those people. And maybe some more I might have forgotten here. Let me start with the motivation for PETab. So in systems biology, as you just heard, we usually have models with a priori unknown parameter values, and we need to infer those from measurements, for example, by optimization. So for that, there are many tools available. For example, SBstoat, many others. That's the good news. Every tool has their advantages, disadvantages, and it might sometimes not be clear and you would like to switch between different tools. That's, however, in practice, not that easy. So what we usually have is that every tool really requires their own input format. So you would have to reformat your measurements, redefine your observation model, linking the measurements to the model. And it's not really a fun activity. It's quite error prone. You need to get familiar with all the different formats first. And it's not so helpful. Also, if you look at some published results, it's often very difficult to figure out what has been done in terms of parameter estimations, which data really has been used, how was it linked to the model. And we felt like there is somehow a lack of a clearly defined format that can be used with multiple tools and provides all the required information yeah, you need to solve some parameter estimation problem. So for specifying some models, we have actually great standards. There is SBML, SLML, there is ZML for specifying experimental conditions. All great tools developed in the Combine community, but somehow for parameter estimation, there isn't really anything broadly supported yet. Things might be changing there. The new ZML standard draft also comes with some parameter estimation capabilities, which is pretty nice. But so far there is no really any broad support. That's why we teamed up to develop Ptap. Before I get to the format, I want to briefly introduce a common setup we're facing. Maybe slightly different from what Joe presented before, or a different presentation. Let's say in principle, it's very similar here. We usually have some model of some biological system, some chemical reaction network or whatever. And we have some measurements that we want to infer, based on which we want to infer model parameters. So we want to minimize residuals or maximize likelihood or something to find the most likely or find parameters that can reproduce the measurements. Often we don't have data from only one specific experimental condition, but looking at different knockouts or different cell lines, general after applying different drugs or whatever perturbation, what Joe referred to as suites of models, pretty much. And often we don't really observe in our experiments directly any specific state of the model, but often it's some more simple or complex expressions of some concentrations in the modeled in there, and we need to link the measurements to the model via some observation model. Here demonstrated a simple case of relative measurements. We have some observable y, which is a scaled model state, and this can be arbitrarily complex expressions. And with each observable, we usually have some error model, because all measurements come with uncertainties, and this needs to be reflected to compute a proper likelihood. To account for different experimental conditions, we usually have some specific instances of a general reaction network to reflect changes in input or output parameters that are also encoded separately. This is a relatively common setup we found that one encounters in database modeling studies, and this is what we wanted to cover in PETab, especially like the parameter estimation part, not really in downstream analysis then. And yeah, PETab is based on a number of tab-separated values files and an SBML model. And this specifies all of those inputs I just mentioned. So the model is specified in SBML because we don't want to reinvent the wheel there. It's a great standard that covers many use cases. Then in addition, we have some file defining different experimental conditions. I will go into more details on the next slide, but pretty much for changing certain parameter values to reflect changes in the environment. We have a file describing the observation model, a file that contains measurements, parameters, and optionally some file that provides that can be used to create some result visualizations similar as you saw in the previous presentation. Those files are grouped together in some YAML file that references each of those files. So further information on that is available on our Read the Docs page. And I will now give you a bit of a more detailed introduction to the specific files here and how they might look like. So on top, you see the previous slide, just a general setup. And I would like to point out how that relates to the different files in Ptap. So for the model, things are pretty clear. It's an SBML file. I don't think that needs further introduction in this audience here. The next thing would be defining different, defining the observation model, which is done in this observable table. So for all the different types of measurements, let's say, we're having one row here, starting with some ID, some expression relating model entities to the measurement, like here, for example, just some scaled state, some information on different on the noise distribution that is assumed, and then some noise parameters. So in the case here for normal distribution, this would be the standard deviation, or here the respective scale parameter for a Laplace distribution. Various noise models are supported here. Then to account for the different experimental setups we might have been using, there is this condition table. Again, some ID for the given condition, identifier for your experiment, for your cell line or whatever, and then certain model parameters that were defined in the SBML model can be overwritten here, for example, to account for different initial concentrations or whatever might be in the model. Then we have the actual measurements that we want to use to infer parameters from. And they reference the observable table here via the observable ID, the experimental condition here, and the time is given when the measurement was taken, and the actual value for the measurement. What is not shown here is you would also include things like the observed standard deviation or similar points. Right, and you have one row per measurement here. And these four files are complemented by a parameter table. This pretty much lists the different parameters, whether they are to be estimated or you want to fix them to some specific value, potentially overwriting anything previously defined in the SBML model. Also, you would specify parameter bounds for optimization in this table. This is again not shown here to save some space. Yep, these are the basics here. There are a couple of additional features, all documented on the PETab webpage. Happy to take specific questions there later on, but I think this should give a rough overview at least. Yeah, the files are, except for the XML file here, easily editable with your favorite spreadsheet application. So there is not too much of an obstacle for working with those files. Yep, so much for the format description. So our motivation for creating this whole thing was to increase interoperability of the different tools. And so far it's only one additional standard, which doesn't really help so much. So therefore, with the help of many tool developers, we implemented import and or export functionality in various tools for simulation and parameter estimation. It's currently 9 tools. You may know some of those, Copasi probably being here the most well-known. And yeah, so each of those tools supports PETab at least to some extent. That varies a bit. And it should make life much easier if you would like to try different tools and try out some different algorithms. Tools are implemented in different languages, so it's quite helpful if you don't need to worry about specifying all your observation model and data somehow in code where you don't really know how to do it. And this should keep the required coding to really a minimum. Right. So we have 9 tools currently supported. So this is pretty good. I think before there wasn't really any format supported by a significant number of tools. We'd be very happy if we could extend that list soon. I will add SBstoat as a future candidate here for the next talk. If anybody plans to extend or implement Ptap support in their tool, we'll be very happy to provide assistance there or discuss any potential issues. Also, if you're not a tool developer, we have a couple of resources that you might find helpful if you would like to have a closer look at Ptap or want to use it in your application. Everything is linked on this website here. We have some step-by-step tutorial explaining for one example model and data set, how you would create those Ptap files and why you would write certain things in certain columns there. We have also a bigger collection of not commented example problems. Those are based on more or less recent parameter estimation studies in systems biology, various models, various types of data. On the one hand, they demonstrate that Ptap can be used to represent those problems. And furthermore, this might be a helpful resource if you're developing some new algorithms and would like to run it on a couple of benchmark problems. So here you have everything in one format. You don't need to scrape through 30-page PDF supplements somewhere to find all the required information. It's freely available here and should be rather convenient to use. When setting up your Ptap files, our Ptap validation tool might come in helpful. It checks whether you really follow the format specifications and gives you some hints on how to fix certain issues. It's a command line tool. It's included in the Python Ptap library, which is available via PyPy. In addition to the validation tool, this also contains some functions for visualization of model fit or also just data, and some functions to package Ptap files into combine archives. For tool developers, again, we have a test suite with currently something like 17 test cases, I think, with some tiny model and illustrating the different features of Ptap and providing some expected values for model simulation. And this might be very helpful to check your implementation and also continuously validate the implementation. Right. All those resources are available under permissive licenses, and yeah, there should be linked on the website here, as I said. Right. With this, I want to talk a bit about the development of Ptap. So we hope that Ptap is developed further in a community effort. We're really happy about any suggestions for additional features, any comments. Most of the development is handled via GitHub, so just post some issue there, join the discussion. Very happy about that. Any future versions and format changes will be discussed there, and the community will vote on whether certain proposals will make it into the next version of Ptap. This is more detailed. The process is more detailed on the website. I will not go into too much detail. The whole development is overseen by an editorial board consisting of 5 representatives. These are currently Jan Hasenauer, myself, Frank Bergmann, Svenja Kemmer, and Fabian Fröhlich. This is the first editorial board now. In general, editors are elected by the community for 3 years. For the first round, some people were only elected for 1 or 2 years to avoid replacing everybody at once and to have some continuity. Right. I want to highlight a few extensions to Ptap we're currently working on, which some of you might find helpful maybe. The first one would be that we would like to extend Ptap to allow for specifying model selection problems. So in many cases, you don't really have one specific model, but you have several competing models that you would want to like compare to your data and see which model would actually match the observations best. And so this is very close to like what I presented before, or what Joe presented before as parameter estimation. But here we would like to add support for specifying a set of candidate models and making it easy to specifying a potential large number of candidate models and running model selection in different tools. It's still at draft stage, but I think it's we're aiming to finalize that in within the next, let's say, 3 months. Things are available online. If somebody's interested, I'm happy to share some further information. Another extension would be about supporting rule-based models. So far, as I mentioned, we only support SBML models. In many cases, this might not be practical. If you have a rule-based model, you can convert it to SBML or to classical SBML, let's say, but things might not be too handy. So we would like to support, for example, PySB or Biosimulations language models here as well. Another point is we would like to integrate Ptap into the Biosimulators and Biosimulations framework. So for those not familiar with that, like Biosimulators database or repository contains a number of tools like packaged with some coherent interface to run model simulations. So you can access many different tools through one interface. You can run model simulations. It would be amazing to extend that also to parameter estimation. And we're discussing next steps there, or have some initial plan. And one part would be providing one coherent interface there. The other part would be actually allowing through the Biosimulations platform, actually running parameter estimation tasks also in the cloud. Further discussions, which are at a pretty early stage, evolve around parameter estimation result format. So would be pretty cool to have some coherent format where you can share and publish your parameter estimation results in some machine readable and understandable and well annotated way. This would be super useful, but we didn't get too far there yet. And yeah, I'd be happy to hear what else you might find helpful. That's already the end. So to summarize briefly, Ptap aims at specifying parameter estimation problems in a more reusable format, and it's currently supported by 9 tools already. Hopefully more to follow in the future. And I mentioned a couple of hopefully helpful resources we made available together with this standard. And you can again find a couple of links here to the documentation, Python library, manuscript we had recently published in PLOS Computational Biology. And right, we also have a Google group or mailing list. Feel free to subscribe. Happy to have more people there. It's actually pretty low traffic currently, so please join. Thank you.
Thank you, Daniel, for a really great talk and giving us a better understanding of the PETab format and sort of the gap it seeks to fill in terms of systems biology standards. We're going to now open the floor to any questions. I know there was a question in the chat for Joe specifically, so maybe I'll read that first to get us started, and then we can take more questions. Okay, so Daniel Zuckerman says, for Dr. Hellerstein, one comment: cross-validation based on alternating time points seems dangerous because such data sets are not necessarily significantly correlated. Fully independent data folds would be much better. A related point in question: to what extent does your pipeline account for the full landscape complexity, multiple minima, you explained so nicely? Is the analysis pipeline only self-consistent insofar as the local features near the best discovered fit are concerned, or would the analysis be reproduced with an entirely different set of starting points, random seeds for exploring the global landscape? He thinks it's an important issue, and a simple heuristic might be to rerun the entire pipeline several times to check for variation.
So let me start with the first question. The reason, and this is something that I don't currently parameterize, but the choice of folds is something that could be available for external parameterization. But the default of alternating time sequences was because we're trying to fit a time course, and the dynamics will vary over time. And if by chance you chose for one fold all steady state values, you would get very poor kinds of fitting results. But I understand what you're saying. Yeah, obviously there are time serial correlations. And so it may be that for some applications, different selection criteria should be used, and this could be something that was given as an option. For the second part, I think you're really talking about this broader issue of fitting it as an exploratory data analysis. And yeah, I agree. It's very true. You do different restarts, you can come up with different fitting results. So I think this is all part of a broader thought of, for reaction networks, what are good fitting strategies? Because it is an exploration, and it probably depends on the nature of the network itself.
Yeah, just to put maybe a finer point on that. I mean, one, we're all data people, right? And when we see that data file with a sort of maybe not definitive, but it looks definitive when you've got your chi-square and those numbers there, and you're like, okay, I did my due diligence, I'm done, I'm reporting this. I worry that might give a false sense of security to people, and I liked your emphasis on visualizing stuff, and yeah, that's entirely on target, I think. But it's just I think that rerunning our pipelines, presuming there's some stochasticity in them, is going to be very valuable for everyone, right? Worst case, you get the same numbers, and you're like, okay, I'm good. But I think we know that's not going to happen, and that would be the warning.
So I have a question for Daniel. I'm sort of curious broadly for PETab, what is the broader vision? I mean, the initial part, which is very compelling argument to me, is to standardize the set of inputs for parameter fitting. I think that's excellent, very much needed. But towards the end in your talk, and you talked about the future, about model selection problems, this gets PETab more into the space of potentially even workflows, where underneath you have some capability for parameter fitting. You're applying this iteratively in many ways. I mean, you could argue if you're going to do that, then you could do bootstrapping too. You could do cross-validation as well. I mean, that could all be workflows within PETab. So I'm trying to get a sense of, the vision for PETab.
Yeah, thanks. Excellent question. Yeah. So yeah, Ptap is so far mostly about, it is about specifying parameter estimation inputs, as you said. For the question whether we're aiming for some like workflow definition, I wouldn't say so. It's more about, again, the input specifying, for example, candidate models, which is a very thin layer on top of specifying those parameter estimation problems. It's again changing a couple of parameters, for example, or specifying multiple models. We would not really go into specific algorithmic details, like how you would want to perform model selection. Again, for the parameter estimation part, we're also not really specifying which optimization algorithm you would want to use, which specific simulation algorithm you would want to use, which I guess one would require for really a workflow definition. But yeah, for some additional features or potentially helpful things, I guess we would have to decide whether it really still is PETab, or that would be more like some spin-off or yeah. I hope that answers your question.
All right, Herbert and Matthias both have questions, so let's go with Herbert first.
Oh yeah, okay, thanks, Monica. Yeah, these are both really nice talks. I assume you can hear me, right? I got some mic headphone issues this morning. But anyway, yes, I'm interested in PETab. One question I have is, I mean, there are a lot of tables, and I quickly looked at the GitHub site, and you've got software there. I'm wondering whether you have software that quickly generates the tables, say default tables in YAML and a YAML file without having to do this manually. Maybe I don't have any information on the noise distribution. Maybe I don't have any observables. I'm just looking at the state variables themselves, etc. Are there shortcut methods I can just call to create a sort of default setup?
Partially. Good point. So there is within the Ptap library, there are functions, for example, for creating those parameter tables. Or maybe do you still hear us?
Okay, no, yeah, there's something wrong with the headphones. When I put them on, they go off.
Okay, so yeah, there are some functions in the Ptap library that can help you with that. It's something I personally would like to extend to allow for more like interactive composition and for like minimal input to specify those problems. There we can still simplify things. There is actually another Python package called YAML to SBML or YAML to Ptap, which allows you specify in one single file both the model and the data and number of parameters, which is like pretty easy to use if you just have a tiny model, and then that's great. If your data set or model gets more comprehensive, then manually writing such a file might not be super helpful.
I mean, one of our target objectives with SBstoat is to make it really easy for new people to use. So maybe this is something we can help as well. If we implement PETab, we can create a really easy interface for people just to, especially for simple models where they don't need all the other stuff. They quickly get it going. Yeah. Anyway, nice work. Thanks.
Thanks.
Yeah. Hi. One question I always had, like, how reproducible are parameter fitting results? And now for the first time, you have here a large set of model problems and 9 tools to implement that. For me, the first thing I would do is like run all the tools over the different problems and see what different parameter values do I get? How big is this problem with the local parameter fitting? Perhaps you could give some insight. Was this done ever, or is this planned? And
yes. There was a paper from 2-3 years ago. I will share the link later on, which used the previous version of this benchmark collection, or which originally published some benchmark collection and ran various optimization algorithms on certain tools. Now, by reproducibility, you mean like in how many cases really the exactly same parameter values are recovered?
How often you get like similar good fits with different parameters? How often you get really similar parameters? How does it depend on the problem size and the kind of problem? Things like that.
I think in most systems biology applications, you have non-identifiable parameters, so you will potentially have arbitrarily many parameter values describing the data equally well. it depends a lot on which optimizer you're using. Some get stuck in local optima more easily than others, or some don't even converge to local optima, but just exit prematurely. So you will have a wide variety of results. So while we're always doing some multi-start local optimizations, this might still not be enough, but we're trying not running optimization once, but really depending on the problem, like ideally a couple of hundreds or thousands of times to get some confidence that we find really a good fit.
Yeah, thanks for the nice answer. Probably we also need something like this, ensemble methods, and just have to run everything and combine the results of all the things. But PETab could enable that, so yeah.
So I have a question for Joe mostly, but both of you. By the way, great talks. Any thoughts on stochastic modeling? If your model itself is stochastic, then your results obviously are not going to be the same every time. So how do you fit with that thing? Any ideas on that?
I'm not sure that there's any great insights there. I think that since, even the deterministic problem is really an exploratory analysis, I think, it obviously carries over to stochastic, and, even for the deterministic case, you may very well use stochastic algorithms to explore the space. So my intuition would be, and to be honest, I don't do that much stochastic modeling. It's primarily been deterministic. But my intuition would be that the approaches are fundamentally the same. What I think we really lack in the field right now is sort of some quantitative sense about the limits of what we've got in the way of results. Because of identifiability issues, overfitting issues. And then with stochastic, of course, we add on top of that, sort of the noise in the model itself. It's not noise in the observational data, it's noise in the model. So I think there's a lot of gap there that we have in terms of our methodologies.
Right. I wonder if you could say, could fit the chi-square space maybe, rather than the gradient descent, for example. So you don't trust any single model result, just plain say, let's fit our model results in chi-square space. I wonder if that's done or possible, or
it could be. I mean, it's an interesting thought. I mean, sort of the avenue I've been in the early stages of exploring is trying to understand the relationship between the reaction network and then the fitting surface. Mm-hmm. And so that I have more of a sense about, the landscape. Am I going across, a super highway, or am I climbing, some very complicated mountain, and I'm going to get stuck in valleys? Yeah, that's sort of been my approach right now, but I think there are a lot of avenues to explore.
Thanks.
All right, it looks like we are just out of time. Thank you, Dr. Hellerstein and Dr. Weindl, again, for your great insights on parameter estimation and your very useful tools. We hope you will join us next month. We're going to have an adjusted time schedule. So our talk will actually be at 1 p.m. PDT, which isn't the best for our colleagues in Europe, but we're trying to accommodate a speaker from New Zealand. So we'll hear about an annotation package. So we hope you'll join us back for that. Thank you, everyone, for joining us today.